The overall goal of the following experiment is to purify at once and at high purity a wide range of meiotic cells from mouse adult testes in order to study the dynamic processes taking place during this unique cell cycle. This is achieved by efficiently dissociating testicular cells with a series of enzymatic digestions with collagenase and trypsin in the presence of DNase I to obtain a single-cell testis suspension. As a second step, the testis suspension is stained with the Hoch's 33342 fluorescent live DNA dye, which allows various meiotic cell types to be distinguished based on their DNA content and density. Next, meiotic cells are analyzed by fluorescence-activated cell sorting in order to identify and purify all the necessary fractions for further analysis, such as cytogenetic or gene expression analysis. Results are obtained that show efficient purification of hard-to-get meiotic cell populations at high purity based on comprehensive cytogenetic and expression array studies.
The main advantage of this fax-based protocol over existing methods like centrifugal elutriation or gravity sedimentation is that it allows to collect at once most meiotic fractions at very high purity. Individuals new to this method will have to combine very different skill sets to perform efficient testis dissociation and staining, followed by optimal cell sorting. Good interactions between the person preparing the cells and the flow cytometry operator, as well as a good understanding of the fundamental of this protocol, are essential for success. Demonstrating the cell dissociation protocol will be Dr. Irina Getun, a senior postdoc in our laboratory who has considerably improved this protocol. Bivian Torres, our flow cytometry operator, will walk you through an optimal setup for efficient cell.